Hi, everyone. Welcome to week. Sorry?
Yeah, we are in live.
Yeah, we can start recording.
Yeah, we are in live.
Okay, thank you. Okay. Hello, everyone. Welcome to week 2 session of the live interactive session for NPTEL's basic course in ornithology. Since the pre-recorded lectures for this week, we've been discussing anatomy, physiology, and color. We are honored to have with us Dr. Anusha Shankar. She'll be discussing hummingbird physiology. Dr. Anusha Shankar is an integrative and physiological ecologist who's mainly interested in understanding how animals manage their energetic needs. She's also an assistant professor at TIFR Hyderabad currently, and she's done her PhD from Stony Brook, New York, and her postdoc at the Cornell Lab of Ornithology. So all yours, Dr. Anusha Shankar.
Thank you, Rafisa. Thank you all for being here. I'm looking forward to a fun session. I did this last year and it was a lot of fun and I got a lot of good questions. So I'm excited to see what questions you have for me after this one. I'll just start by sharing my screen. Okay. You can see that, right, Nafisa? All right.
Yes, I can see your screen.
Perfect. So yeah, for the last 11 years or so, I've been studying hummingbirds. And there aren't hummingbirds here in India, but I'll be pivoting to study other species with similar questions as the ones that I used on hummingbirds. And I'm really interested in these animals that can generate their own body heat, which are called endothermic animals like us, but also change their body temperature to save energy. So I'll tell you a little bit more about them. But before that, I'll tell you a little bit more about my background, because it seems like all of you come from very different walks of life and with very, very varied experiences. So just to show you my career trajectory and my life trajectory, I spent a lot of my childhood in Chennai, but we moved a lot when I was a kid. So I went to 10 different schools. I adjusted to new environments all the time. And this is me playing with a camera case when I was, I don't know, 2 years old or something. And this was not me growing up. I have a lot of colleagues who study birds, who grew up with binoculars in their hands and watching birds. And that was definitely not me. I didn't have that much of an interest in birds. I wasn't particularly a super outdoors person. And I really had no idea that you have to choose a career and stick to a life track in general, at least in the Indian context. That's what's expected of you. And you don't just do whatever you want and make a living in general. So I ended up going to do my undergraduate degree in zoology. And I was also really interested in psychology. And I also thought I'd be an actor. And I also thought I'd be a pilot. I just wanted to do a bunch of stuff. And I didn't know why you had to choose to do just one thing. I still feel like that sometimes. But I ended up doing internships just to test out what I really liked doing. And I've spent a lot of time in the field. I had a tiny point and shoot camera. So I spent a lot of my time looking down, unlike birders who look up. And I looked at little insects and snakes and took photos of things that I could see close up. And then one of my earliest experiences in wildlife biology was a project that I did in Assam with the Wildlife Trust of India to track a hoolock gibbon. So I was studying this female hoolock gibbon's behavior when she was being rehabilitated into the wild. Her parents had been poached, killed by poachers, and so she was orphaned. And after spending 4 years in captivity, they were trying to rehabilitate her into the wild. And then again, I took a lot of photos of tiny things. And then I did another internship to track king cobras at Agumbe in Karnataka. So they put radio trackers on these giant king cobras, and we were following them to see what their habitat use was. And if you relocate them, do they come back to their home grounds and things like that. And then I ended up doing a master's in ecology, and I studied these beautiful birds called hornbills. If you've seen them, just like put a thumbs up in the chat. They're really huge, and they nest in tree cavities, and the female goes inside a hole in a tree and takes care of her chicks and her eggs until they hatch, and she doesn't come out of the nest for 1.5 to 3 months, depending on the species. So these females have an extreme, life history where they completely depend on the male to bring them food when they, sorry, let me just Can I mute this person? Do you mind? Yeah. So they completely nest in these tree cavities, and the male brings them food, and they have no other source of food when they're nesting. So I was studying how these females choose a nest site in a very fragmented human-dominated landscape in Maharashtra, in the Western Ghats. And then at each stage, I didn't know what I was going to do next. And it's a lot of angst and a lot of career decision-making and things like that, which never ends. But I ended up going for a PhD to Stony Brook in the US in New York. And while I was there, I spent a lot of time studying hummingbirds. So this was my first experience, holding birds, catching birds, studying something deeper than their ecology. And I went to Arizona, which is in the southwestern US, close to where you are in California. And studied these hummingbirds in this kind of desert landscape where they feed on these tiny little red flowers that you might be able to see in the foreground. I spent about a year doing fieldwork in South America, in Ecuador. So it's right on the equator, a tiny little country, huge hummingbird diversity. And I spent some time in the high Andes, in the high elevation mountains, and also in the mid elevation Andean mountains. So there's cloud forests in the mid elevation, and in the high elevation, it's kind of the short, scrubby kind of habitat. And so I morphed slowly from doing ecology in my master's to physiological ecology in my PhD. And I've been moving steadily towards becoming a more integrative biologist because I'm really interested in what organisms do. And how they interact with their environment, but also what goes on inside their own bodies. And I'll talk to you a bit more about that. So broadly, I really am interested in the mechanisms that animals use to cope with environments that are different and environments that change. And hummingbirds are a great way to study this. Hopefully this video is showing for you, but if you haven't seen a hummingbird, they're extremely fast, very small. They use up energy very quickly. And they mostly get their energy from high energy nectar or sugar water from plants. So this is an artificial feeder in the cloud forests in Ecuador. And there's like 20 different species of hummingbirds at this one site. And many of them are at this feeder. So they use up energy so fast. And that makes them interesting because they barely store any fat. So they don't have much of a backup source of energy. And if they don't keep feeding to match their metabolism, they'll die. So just to give you a sense of how extreme that is, I made this graphic. Let me just use my laser pointer to make sure that you can see. So an average human would, if they could just feed on potato chips the whole day, they would need to eat about 15 packets of potato chips every day to meet their caloric needs. And if they had the metabolism of an elephant, but stayed the size of a human, they would need only about 4 packets of potato chips a day to meet their daily energetic needs. Can you guess how many packets they would need if they had the metabolism of a hummingbird? Can we put that in the chat? If a human had the metabolic rate of a hummingbird, how many packets of potato chips would they have to eat every day? Oh, okay, and people have seen hornbills. That's good to know. Okay, so we have guesses, 100, 1,000, 350, 50. More than we need as a human. That's a good one. Yes, 36, very specific. 15, also very specific, yes. 60. Okay, we started high and we're going low. It's very interesting to see. Okay, I think I'll give you the answer. So hummingbirds we'd need about 600 packets of potato chips. And this is just an average. So for some hummingbirds that are spending more energy in a day, we need like 1,200 packets to meet their daily energetic needs. So they're using up energy very fast per gram of their body's mass, right? Much faster than we are. And they don't have much of fat stored to stay alive. And yet they manage to live in such a huge diversity of habitats. They live in the cloud forest, like I said, in the deserts in Arizona, in the high elevation Andes in Ecuador. And they're able to survive in all of these different environments with the food and the temperature and the rainfall and all the differences that come with that. So on the way, while I was studying all these different environments, I was also meeting a lot of diverse people and getting some really interesting interactions with biologists and non-biologists who love birds. But yeah, back to hummingbirds. So they somehow living in these all these different environments and with those tight energetic needs, they have diversified into over 370 species now that have been classified. And they have such a diversity of form and color and size. There's these crimson topazes, which are super colorful. The tiniest, which is a bee hummingbird, it's endemic or only found in Cuba. And it weighs the same as an old 10 paisa coin. Or in the US, it weighs a little bit less than a dime. So these are tiny, tiny birds. And the average hummingbird weighs less than I don't know, like a 1 rupee coin or something, or a 2 rupee coin, or the same as a US quarter. So this is a giant hummingbird, and it weighs about 20 grams. So it's the biggest hummingbird. It's 9 times the smallest hummingbird's weight. There's the sword-billed hummingbird, which has a beak longer than its body. And then this is one of my favorites. This tiny one on the bottom left is a booted racket-tail. It has these two long feathers. This is a male. And these little boots on its feet, these little white boots. And it flits around in the cloud forests in Ecuador, for instance. So to tell you a little bit about their evolutionary history, it seems like their ancestor was in Europe. About 30 to 35 million years ago, a fossil that was found in Germany was dated to that time. And that seems to be a hummingbird ancestor. And it seems like they came from Europe somehow, we don't know how, through North America to South America, and then the Andes uplifted about 20 million years ago, and they were starting to diversify up the Andean mountain range. And so they hugely diversified. They went extinct everywhere else in North America and Europe. And then from South America, they recolonized North America. And so there's about 16 species in the US, for instance, but 370 plus species total in North and South America. Most of them are in the Andes. There's lots in Colombia and in Ecuador in South America. So there's just so many species with so many forms and functions. And I was interested in a bunch of questions with them. One is looking at their allometry. So they have unusually high scaling slopes, and I'll talk about that in a minute. I was also really interested in their daily energy expenditure, because I told you they spend a lot of energy in a day, but I wanted to know how much of it is fixed and how much of it can vary, but depending on what's happening in the environment. And then a lot of older studies in the 70s found that hummingbirds were mostly spending their time perching. They were spending 75% of their day sitting on a branch somewhere, and only the rest of the 25% hovering and flying. This is from watching them with binoculars. So I wanted to know if we could model that and budget their energetics more, fine scale. And I wanted to know what they do at night, and we'll talk about that in a bit. But there's a bunch of different scales that I would like you to think about in the context of studying organisms like birds. You can study the individual, you can study the species, you can study across species, and you can learn so many different things about biology by looking across these scales. And then you can look within the individual, which I'll also talk about a bit. So the first thing, allometry, is this idea that if you plot on a plot one trait or one sort of feature of the organism against another trait, like its body mass very commonly, then you can try to find if there's relationships that link those two traits that are true for all of life, or for all of one taxon, or for whatever group you want. And so in the early 1800s, people started wondering if there was some rule that governed all of life that could link the energy expenditure, how much energy you spend or consume in a day, with your body mass. And they were starting to find that it seemed like there might be some sort of universal law that links the energy expenditure and body mass in some ratio of 0.75 or 3/4. But then slowly in the 1900s, late 1900s and early 2000s, people were starting to separate these groups out by the taxon. So you plot the birds, mammals, reptiles separately, for instance, and they had slightly different relationships. And so there's lots of groups still trying to find out if there's some way that we can, find how these traits are linked, how energy expenditure and mass are linked. But you can do this for brain size, you can do this for arm length, you can do it for literally any trait that you can imagine, and plot it against body mass and try to find these relationships and find why those relationships might be true. So what do they mean? For instance, if you have a slope of this line of 1, that means that per unit increase in per unit increase in body mass, you have 1 unit increase in energy expenditure. And so a big animal is spending as much energy per unit mass as a small animal if the slope is 1. If the slope is less than 1, like it is for lizards, then a big lizard is spending a little bit less energy per unit mass than a small lizard. Okay. And then mammals have an even lower slope, which is close to that universal kind of slope that they thought existed. It's at 0.75. And then birds have an even lower slope. So actually a big bird is spending much less energy per unit body mass than a small bird is spending. And hummingbirds are birds, so we imagine that maybe they have a slope close to 0.67. But actually in 1999, Nagy et al., they reviewed a bunch of literature and they put together values from other people's studies, and they found that this slope was about 1.21. And this is crazy because it means that a large hummingbird is spending much more energy per unit body mass than a small hummingbird. And that doesn't usually happen in biology. It doesn't happen even in economics. If you have a bag factory that's producing a bunch of bags, if they're producing 10 bags every month or something, and they want to scale up to producing 1,000 bags, they want each bag to cost less to make, right? If each bag is more expensive, then it doesn't make sense to scale it up. You want to stay small then. So just like that with the hummingbirds, why would you bother being a big hummingbird if it's so much more expensive per unit body mass to fuel yourself to be a big hummingbird? So we went out and decided to collect some more data from more species, because that study only had 6 of the species and from very limited groups within the hummingbirds. But how do you do that? You can't put a Fitbit on them to measure their daily energy expenditure. They are way too small, it would fall off. And so we had to find other ways to study how much energy they spend in a day. And so we can catch them with a lot of training and a lot of permits from the wildlife departments, for example. You need a banding permit to be able to put a band on them that can identify the individual. But we use these feeder traps to catch the bird. And this is what they look like. So you can see that iridescence on their throat, which is the color that changes depending on the angle you look at it in. A lot of birds have iridescent feathers like this. So there's a blue-throated mountain gem from Arizona on the left and a black-chinned hummingbird on the right. And you can see it's black sometimes and it's purple sometimes, the throat. Whereas the one on the left was gray sometimes and blue sometimes on the throat. So one way we can study their energetics is to use this doubly labeled water method, which is an ingenious method, I think, where I'm just going to check the chart for a second. Mass is inversely proportional to the energy spent. Okay, we'll get back to this in a minute with the allometry stuff. So the doubly labeled water method, I think, is genius because you can use a double isotope of water, which has deuterium and oxygen 18, and mix it with their nectar and feed them with it. Another way is to inject them with it, but it's a stable isotope, so it's not radioactive, it won't harm them. And you feed them with it and then let that mix with their body's water and collect a urine sample or a pee sample, and then you let them go. And in 24 hours, you try to catch them again and get another urine sample. But while they're out in the wild, they're breathing out carbon dioxide and they're peeing out water. So you can see they're losing oxygen 2 ways. They're losing oxygen in their breath and in their pee, whereas they're losing the hydrogen only in their pee. And so the rate at which these 2 isotopes are lost from the body, that difference tells you how much carbon dioxide they've been breathing out in 24 hours. And that's a really good measure of their energy expenditure, because all of our metabolic processes that take energy, the end product is carbon dioxide. And so we can estimate how much energy they've spent while they were in the wild over 24 hours. And we can do this for many different individuals in different habitats and different landscapes. So we did that for 17 different, we did it for about 12 different species and added the work that other people had done before and plotted it like this to look across different groups. And we found a slope that was closer to 0.96. So this is still almost 1, and the error estimates include 1, but it's not as high as 1.21, which people found earlier. So it's still puzzling a bit. It's not like all the other birds. They still seem to have a slope that's very high, closer to what lizards have actually than to what other birds have. And so I was racking my brains to try to understand how can I understand this better? Why would they have such a high slope across species? And then I found this series of papers by someone named Doug Glazier, who published something called the metabolic level boundaries hypothesis. And what he was suggesting was that your slope kind of depends on what you're spending energy on. If you're spending energy on heat-related things or water-related things, then it relates to your body's surface area to volume ratio. And that's closer to 2/3 than if you're spending on high-power energetic activity, like muscle activity, or things like torpor, which we'll talk about later, when your slope is closer to 1. So I was wondering if things like this could help us understand the scaling slope in hummingbirds. And then on the way, I was involved in this review paper that we published, which was so interesting. This first author, Umat, he drew these illustrations. So he pointed out that there's species in which this is all just one species on the left. And within this species of beetle, of weevil, these are males on top and females on the bottom. And within the species, there's so much size variation that there's some females that are tiny, some males that are tiny, some males that are huge, some females that are huge. And so he was wondering if there could be sex-specific changes in allometric relationships. So not even across species, just within species across sexes, if there could be different allometric relationships. And he was suggesting that you have to take the sex of the individual into account when you're considering these energetic relationships. Because imagine a peacock, a male peacock with a huge train versus a female peacock, or a male elk with huge antlers versus a female elk, or these crabs that have big display claws. Do they have different allometric relationships across sexes? Okay, so back to the daily energy expenditures. I was wondering, how variable are the daily energy expenditures? So we know that the scaling slope is really high. But if we look at each of those daily energy expenditure points, how much do they vary within a species? And so we went to 2 different sites in Arizona. There's one site that's called Harshaw Creek, which is very elevationally and vegetationally diverse. There's lots of little micro habitats like this, where this was a student named Rebecca. She was taking shelter in the shade, and the hummingbirds could do the same. And then another nearby site, which is called Sonoita Creek, where those tiny little red flowers in the foreground are there. This is a much flatter, hotter, drier, elevationally, flatter site. And so we were wondering how energy expenditure could change in these 2 very different habitats. And so we did that same doubly labeled water method. We collected pea samples. And on the left, you can see Harshaw Creek. So this is in the pre-monsoon and the early monsoon, just after the rain started. And Sonoita Creek is on the right with the pre-monsoon and the early monsoon. And so again, Harshaw is the elevationally, vegetationally diverse site. Sonoita is the hot, dry, flat, wet, not wet site. And so these are the data we have. Each black point is an individual hummingbird's daily energy expenditure. And the colored points are individuals that we got in 2 different seasons. So we could see how an individual's energy needs changed in 2 different seasons. And so some of them maintain their same energy expenditure, but some individuals hugely increase the amount of energy they spent when the season shifted from early pre-monsoon to early monsoon. But what was most interesting about this graph is these points in Sonoita Creek. So all these individuals spent some amount of energy, but after the monsoon, they spent hugely increased amounts of energy. So their daily energy expenditure was very variable. They weren't even overlapping in their energy expenditure in these 2 bars, right? I'll just check the chat. Okay. More administrative questions than science, so I'll skip ahead. So I wanted to know then, what is causing this change in energy expenditure? Why is it changing so dramatically between sites and between seasons? And so we have to look at what they're spending energy on to be able to answer that question. And so that comes in, and here comes in a different method. This is called a respirometry system. And it has an oxygen sensor and a carbon dioxide sensor. And what we can do is we can pull the bird's breath while it's doing different activities and see how much energy it's spending, because we can measure the carbon dioxide in its breath. And I already told you carbon dioxide is a good proxy for energy spent. And so is oxygen consumed. They are 2 sides of the same coin. So we can use this machine to measure their energy expenditure while they're hovering, like in this video, or while they're sitting still, while they're at different temperatures, maintaining a constant body temperature overnight. Like in every scenario, we can measure how much energy they're spending. And then we can basically model the amount of time they're spending on different activities because again, we don't have a Fitbit to say exactly 70% of the day is this, 20% of the day is that, but we can model different time estimates to arrive at a similar conclusion. And so I'm going to show you a very similar plot to the one I already showed you, but I'll add on some time estimates. So again, daily energy expenditures on this y-axis, and those sites and seasons are here on the x-axis. And now we'll add on the time energy budgets. So the literature I told you said that they were spending about 70% of their day perching and 30% of their day, I said 75-25, but it was closer to 70-30. 15% hovering, 15% flying, 70% perching is what the people with the binoculars estimated that hummingbirds were doing. But I also modeled a much lower energy scenario where they were spending 75% perching, 25% hovering, flying, and a much more energy intensive model where they were spending only 20% of their day perching, but 80% hovering and flying. So that's much higher. And so the different colors are the energy budget models that I constructed based on the respirometry and on my modeling, the time budget. And the gray bars are the doubly labeled water measurements I already have. So I know that this is how much the total should be in the gray bar. And the colored ones are my model estimates. So many different of these colored scenarios can match what the daily energy expenditure was in Harshaw Creek in the pre-monsoon. But only one situation, the highest energy model, could match what was happening in Sonoita in the early monsoon. So that means that in some situations, hummingbirds are spending only 25 or 30% of their day hovering and flying and most of their day perching. But there's some situations like in Sonoita Creek in the early monsoon, where they have to be spending about 80% of their day hovering and flying and only 20% perching. So that's a huge amount of activity. This means that here they're spending maybe 1 to 3 hours of their day hovering and flying, which is like my activity level, right? I don't spend more than an hour a day on exercise. But here they're spending 13 hours a day hovering and flying and only 20% of the day perching. So I wanted to know what is causing this difference. What's happening in the environment that's causing such a huge shift in their energy budget and their time budget? And it turns out that it's not temperature, it's not daylight, it's not so many of the other things I expected. It's really just down to the flowering. And so in Harshaw Creek, in the pre-monsoon, there were so many flowers. They were abundant, they were clustered, they were dense, they were all in like one place. Whereas in Sonoita Creek and in the post-monsoon, just after the rains, the flower densities actually went down and the flower numbers went down. And so they were more dispersed and there were less flowers available. So it seems like the hummingbirds had to spend more time finding and getting between the flowers after the monsoon had started than before the monsoon. And so the environment really affected their energy and time budgeting. So around this time, I was in the like 4th, 5th year of my PhD, and I was wondering what I was doing with my life and wondering if I was working hard enough or not. And so I started tracking my own time while I was tracking hummingbird time. And I found, like I said, that I spend about an hour or less a day on average on exercise, most of it salsa dancing, whereas the hummingbirds are spending 3 to 13 hours every single day hovering and flying. And so it's just amazing that they can spend that much on really intense muscular activity. So around this time, after I finished graduating, I moved to Alaska, which is in the far north above Canada of the US. And I studied seasonal depression. So this is what it looks like for like 7 or 8 months in a year. And a lot of people get very depressed because there's only 4 hours of daylight in the winter compared to 20 hours of daylight in the summer. So there's a huge shift in daylight. And this causes seasonal depression in like 9% of women in Alaska compared to 3% in the rest of the US. And so we were trying to develop this animal model for seasonal depression, which is called the Nile grass rat. It's a daytime active or diurnal rat, unlike most of the lab rats and mice which we use, which are nocturnal. So it doesn't really make sense to use them as a model for how daylight changes your mood, whereas these diurnal animals are good for that. So we were studying how the light availability and the timing of it changes sleep, changes sugar consumption, and changes daytime activity patterns, as well as things like liver fat in these Nile grass rats. And the gut microbiome, sorry. We also looked at the microbes in their gut. I was really wondering, and so this is a tropical rat that's being used in a lab situation that's very temperate and that's very, extremely seasonal. And I was wondering if I, as a tropical animal, would also experience seasonal depression in Alaska in a temperate environment. And actually, the summers were a lot harder for me. When the day just never ends and you keep going and going and going, that was much harder for me than the winter. But this experience really got me to look at how animals might be used in a very biomedical situation and thinking about what happens inside an animal. And so then I went on to do a postdoc at Cornell, like Nafisa mentioned, where I went back to studying hummingbirds. But that postdoc really helped me think that I could scale from whole animal energetic needs and how they related to the environment to what was happening inside the organism, how the organ level metabolic traits are changing, and what may be the underlying, genetic mechanisms that scale up to that interaction with the environment. And so for most of the rest of this, we'll focus on how hummingbirds are heterotherms, which is that they can change from being endothermic or maintaining a high body temperature during the day to ectothermic or maintaining a low body temperature at night. And I'm fascinated that this exists because in the dark, when hummingbirds are asleep at night, they can't see the food, and so they don't feed, but their energy expenditure would still be high. So to lower their energetic needs, they go into this state called torpor. And torpor is kind of a generic word for including things like hibernation. So you've heard of hibernation maybe that bears do or that ground squirrels do. And that's an extreme version, which is multi-day or multi-week torpor, where you can stay in that state for 6 months, 8 months in a year. But hummingbirds are able to do this on a nightly basis to save energy. And so they're, let me hope that this works. Oh, okay, sorry. This is the computer problem that I was hoping I could avoid. Let's imagine that this is a video instead of a picture, because it should be. And this exists on YouTube if you want to find it. Hummingbird Torpor, Anusha Shankar should give you this video. But this is a hummingbird with a thermal camera. And this is the eye, which is at a high body temperature. And this is the beak, and this is the tail. And so at night, they're breathing and their heart rate is pretty high. Their breathing rate is very fast, and they're maintaining a high body temperature. And they're asleep. The eye is closed in this image. And later at same night, that same individual went into a state of torpor. So the whole body gets super cold. It's the same temperature as the outside air. It's basically becoming an ectotherm, or like a lizard or a snake that allows the outside air to decide its body temperature. And in this case, the bird is at about 15 or 16 degrees Celsius. But in some cases, they've been measured to go down to 3 degrees Celsius. So this bird is like near freezing. And if you look at the video, it's barely breathing. Its heart is barely beating. It's very sporadic. It's like not breathing for 10 seconds, then it takes 4 deep breaths, and then it stops breathing again. Whereas on the left, they're like, so it's a very huge shift. And what this means is that normally humans can maintain a high body temperature, right? 37 degrees Celsius. If we drop by more than 2 degrees Celsius, we're in trouble. It's called hypothermia. We need to be rewarmed externally. Whereas hummingbirds can go from 41 degrees Celsius down to 3 degrees Celsius with a huge 36 degrees Celsius change, and wake up the next morning like nothing happened. They're fine. So they're saving a lot of energy by doing this. They're down to about 30% of their normal metabolism, meaning they're saving like 80 to 90% of normal metabolism by being in this state. And I'll skip this a little bit for now. I think I'm a little slower than I usually am. I'll just go to this. So we thought we could measure hummingbirds at night while they were using torpor or not torpor. And so we use this respirometry system that I told you about earlier. This is with Emily, who's at George Fox University. She just graduated. But we thought that we could look more at what was happening inside the organism. And so we built these chambers where we could measure both the metabolic rate with the respirometry system, and also use the thermal cameras to look at surface body temperatures of the bird. Because earlier, our chamber allowed us to do either one or the other. And so these chambers that we built, we replaced this front panel with a thermal window. that's opaque to humans, but the thermal camera can see through so that it can get an accurate picture of body surface temperature. And so we were able to image the hummingbirds like this and see when they went into torpor. And our goal was to study gene expression in the hummingbirds while they were in torpor. And so we had respirometry at the same time as thermal imaging. And then we also collected tissue samples while they were in different metabolic states to look at gene expression. And unfortunately, to do this, we had to kill the hummingbirds. But it's very few individuals, and it's a species that's increasing in population size. And so we had to go through a lot of permitting to do this and a lot of animal care and ethics approvals. But there's really no other way to study what's happening inside the organism other than to do this. And I was really interested in it because when these hummingbirds go down into torpor, all of their metabolic functions are slowing down. And we realized when we were dissecting these organs out that the heart keeps beating in torpor for like 10 minutes after the bird has been killed. Whereas when it's not in torpor at a high body temperature, when it's asleep, the heartbeat stops beating almost instantly. So there's a lot of biomedical potential to understanding how this works. So if we don't do that, if they stay in torpor the whole night and they arouse at the end of the night, then they come back to normal. Their heart keeps beating normally. It's not arrhythmic. It's not tachycardic or bradycardic. Their brain functions normally. They still remember where the flowers are. They still remember who they are, what they have to do in a day. And they can go through this change every single day and still function normally. So it's just such a fascinating strategy. And if they're functioning at 10% of their normal, I wanted to know what is so important that's keeping them alive when they're in that state. Because if you disturb them or if you shake them or anything when they're in torpor, they can't respond at all. They're completely useless for 20 or 30 minutes until they warm back up to normal body temperature. So I wanted to know what's keeping them alive when they're in this state. And so I worked with a couple of undergraduates and a senior scientist at Cornell to look at gene expression in these different tissues across metabolic states. And we found that many genes are downregulated, which means that their expression decreases in torpor. But there are some genes that are upregulated that increase in gene expression when the animals are in torpor. And that's super interesting. One of the big pathways that's upregulated that increases in expression in torpor is the circadian pathway. So it seems like keeping track of time is really important for them, even though they're functioning at 10% or 15% of normal metabolism. And I think that's because they have to wake up about an hour before sunrise. Almost every individual I studied correctly knows, even in the dark, 1 hour before sunrise, that it starts warming up. So keeping track of time seems important, and also probably synchronizing all the body's functions. So all of the organs are working at the same time, doing the same thing, seems to be important for animals in torpor, or at least hummingbirds in torpor. And so, for instance, if you look in the heart or in the lungs, this gene called clock is at some expression level in normothermy, which is sleep, high body temperature, similar levels in when it's entering torpor, but it increases in expression when the animal is in deep torpor. And the same is true for the lungs. And so we saw similar kinds of gene expression patterns in these other genes that are in the circadian pathway called Cry1 and Cry2. And we saw the opposite in these two genes called PER2 and PER3. And these are all implicated in mammal torpor as well. They seem to be important in regulating mammal torpor with slightly different patterns. And so this is the plots for Cry1 and Cry2. But there's a bunch of other pathways like the metabolic pathways, cardiac function, mitochondrial function, which is the energy source of the cell, right? And fat metabolism and storage, which are all interesting pathways that we want to look more into. Then there's other pathways that are downregulated, which is also interesting. There's some tumor suppressing and DNA damage and protein modification pathways that seem to be downregulated or less expressed in torpor. And then I had a student who was looking at the brain, because in mammal brains, there's so much interesting stuff going on in the hypothalamus and the hippocampus and the cerebellum to coordinate torpor, because these brains are also at 5 degrees, 10 degrees Celsius, and then they're reworking the next morning again. So we're also starting to look at mitochondria, because we all read in our school textbooks that mitochondria are the powerhouse of the cell, right? I didn't know that they move and that they change. They're super dynamic organelles. So all these little worm-like things, I saw this on a Twitter thread and I was so fascinated that I didn't know that mitochondria was so cool. But they're very dynamic. And so someone, when I was interviewing at TIFR Hyderabad, suggested that I could use electron microscopy to look at mitochondria in the different tissues while the hummingbird is in torpor and not. So we can circle, this is a skeletal muscle, and we can circle the mitochondria in the skeletal muscle and see if they change in number and size when the bird is in torpor versus in normal high temperature sleep, because their energy expenditure is changing so much. And then there's a bunch of other things we were able to do with these thermal cameras. I worked with this student who now has finished his PhD, but he started with me 8 years ago as a field assistant for 2 weeks in Ecuador. And we worked together on a project to put thermal cameras on these females that were nesting in the wild in California. And so his question was, are these females, the females are the only ones incubating the nest? And so are they going into torpor and prioritizing their own energetic needs? Or are they avoiding torpor and prioritizing keeping their chicks and their babies warm? So he found mostly that they seem to avoid using torpor. There were 2 nights out of the 108 nights that he studied that they did use torpor, and a few other nights where they used the shallow version, just dropping their body temperature maybe a few degrees Celsius. And those nights when they used really deep torpor, there was rain. And so there seemed to be a situation that caused them to, environmental condition that caused them to use torpor, whereas they avoided it on all the other days. And it seems like the nest really helps keep them warm. So their cost of not using torpor is lessened because the nest incubates them as well. So overall, heterothermy is this really interesting strategy to deal with environmental variability. And it's not just cold temperatures. In many situations, if the food is unpredictable or food availability is low, or if the birds are going to prepare for migration and they want to fatten up, they use torpor more. And mammals that in Australia were found to use torpor more when there were forest fires. So there's many different situations, environmental conditions and internal body energetic situations that can cause torpor use. But it's been very understudied. And so less than all now there's like 11,000 described bird species. And we know if torpor exists or not in less than 1% of them. And so some of the things we're doing is looking at torpor across the bird phylogenetic tree. And I have this great undergraduate student at Cornell who is looking across birds and across the bird phylogenetic tree at torpor use. So this is the phylogenetic tree of all the birds, and we've plotted the points where we have data for torpor use. And the colors are how cold they can get. So the cooler colors, the dark blues are more torpor, deeper torpor. And these are all the hummingbirds which get the coldest. And then we did a similar thing for mammals. But my plan over the next, however long I'm doing this, is to look at a broad kind of global framework of heterothermy. I want to look across different groups that are free-living organisms like mammals, birds, reptiles, non-avian reptiles, and look at sleep and torpor. And I think this is really interesting because we have 2 endothermic groups, the mammals and the birds, and we have these ectotherms, the non-avian reptiles. And these 2 are closely related, and this is not as closely related, but these 2 are the endotherms, and this is the ectotherm. And the heterotherms are spread between the mammals and birds. So what is the evolutionary origin of heterothermy? What is the evolutionary origin of endothermy? These are all questions that we can get at by looking across these groups. And I'm really interested in environmental gradients because, like in India, we have so much human land use change, and we don't have enough information of how animals change to adapt to that difference, especially in the tropics, which is why I'm back in India. I really want to work in the tropics to study these environmental effects on animal energetic change and use. And eventually, I would like to study organ-specific gene expression here as well. And those energy budgets that I told you about. And so I think being here, especially in Hyderabad, there's both there's a bunch of species that I'd love to study. So there's some natural experiments that are set up great for this. So this is the bird phylogenetic tree. I'll just check my time. Okay, I'll be done in a few minutes. So this is the bird phylogenetic tree. And here, this big group in the dark blue are the passerines or the perching birds. And if you study the bird phylogenetics later in this course, you might see this, but these are 6,000 out of the 11,000 total bird species. And hummingbirds are somewhere here. They're not passerines. they're close to the nightjars. They're very closely related to nightjars in this tree. And this is a really interesting comparison because hummingbirds and nightjars, despite being so closely related, are ecologically very divergent. They occupy very different ecological niches. The nightjars are like this big, hummingbirds are this small. Nightjars look like that. They kind of camouflage into the ground where they often are found. And they're insect feeding species, whereas hummingbirds are largely nectar feeding species, though they also feed on insects. What's interesting is both groups have been found to use deep torpor. They can lower their body temperature by more than 20 degrees Celsius. And on the other side, you have hummingbirds and sunbirds, which are evolutionarily very distant from each other on opposite ends of this tree, but ecologically convergent, very similar looking, occupy a very similar ecological niche. They both are nectar feeding. They're both small. They both are colorful and they have iridescent feathers. And so they occupy very similar ecological roles from very different evolutionary trajectories. And they're also geographically divided. So the hummingbirds are in the New World or in North and South America over here, whereas the sunbirds are in the Old World in Asia and Africa. So we can also look at evolutionary convergence or divergence by looking at these two comparisons, for instance. Can you hear that knocking, Nafisa? Is it super loud? Okay. So the other interesting thing is that these animals, there's no really good collective data on this, but animals that use torpor seem to have very long lifespans. So hummingbirds, you would predict them to live 1 or 2 years for their body size, but they live an average of 7 years. And there have been some hummingbirds that have been recaptured after 12 years. And so this is true for bats that use torpor as well. They have unusually long lifespans. So there's so many questions about lifespan, about the evolution of heterothermy, evolution of endothermy, but that we can get at by comparing birds and mammals and non-avian reptiles, like I said. There's also a bunch of interesting lessons related to biomedical and molecular mechanisms that people are starting to learn from studying torpor in mammals. So mammals that hibernate seem to accumulate these tau proteins in their brain that when they come out of hibernation, they can clear out of their brain. And humans that have Alzheimer's also accumulate these tau proteins, but don't clear them out of their brains. So how do they do that? How do animals coming out of hibernation get rid of all those tau proteins? And then I told you that the heart rate is super slow in torpor, and then they come back out of torpor and it functions fine. So imagine if we can understand doing this for like preserving organs. If we do a heart transplant and we can slow down the metabolism of the heart, but then bring it back without damage to the heart, that would be really useful in a biomedical context. And then immune function in torpor is suppressed. So there's something called white nose syndrome that's hugely decimating the populations of bats in North America. And it seems to really get them because the immune function is suppressed when they're in hibernation. That's when the fungus infects them and really affects them the worst. So we can understand things about immunity and about how they keep track of time, because I told you the clock genes are upregulated in torpor. They're actually suppressed in hibernation because you don't want a 24-hour rhythm when you're in a weeks-long torpor. So there's lots of interesting implications of studying it. And we could do therapeutic hypothermia. So there's a couple of companies in the US looking into doing this medically, and a bunch of medical researchers that are thinking about this. But if you can cool down at least regions of the body, if not the whole body, like parts of the brain in a stroke patient or the heart in a cardiac patient, when they've had a cardiac attack, then you can slow down metabolic function and therefore slow down damage while the surgeon does their thing. So you can gain time and lessen the damage by using something like therapeutic hypothermia, by cooling down that organ. And then I read a lot of science fiction, so it's interesting to think about human hibernation and sending humans or animals into long-term hibernation for space flight, because you'd save a lot of payload on the spacecraft by not giving them food, by them not producing as much waste, by them not needing as much oxygen, exhaling in as much carbon dioxide. So human hibernation is often used in lots of science fiction, but they don't really get into how that would work. Like, would you be the same human if you came out of hibernation after years? I don't know. Your memory would kind of be weird. So how do animals do that would be a good question. So I'll end in a minute, but I'm really interested in doing science outreach. This is probably like my 112th talk. I love giving talks to the public and interacting with them about hummingbirds. And I love mentoring students. I've done that a lot. And since moving here, I've gotten a lot of requests for people to do internships with me. But I love grant writing, and I love getting grants and writing them. But to give you a sense of the failures that I've had, as well as the successes, I know you're in a stage when many of you at least are probably in a stage when you're applying to further degree programs or thinking about applying for whatever, jobs and studying. So when I applied for PhD programs in the first year, I sent about 8 applications to places in the US and the UK, and I got 8 rejections. So I didn't know how to do it. And I hadn't done it before, and no one was there to really tell me, you have to write to people if you want to go do a PhD in the US. You have to establish a one-on-one relationship with your PhD advisor before you apply. Otherwise, you're not going to get in. So the second year, I found out more. I wrote about 80 emails to different professors, and only about 30 of them responded. But out of those 30, I submitted about 8 applications for PhDs, and then I got into 3, but I was rejected from 5. And so you really can't take failure and rejection personally. You just have to learn as much as you can from it. And then with grants, I've applied to more than 50 grants or awards, and I've gotten about 30. Many of them are very small. So I built up a lot of funding from very small grant sources, but I ended up raising about 3 crores total in the last 11 years to fund my research. It takes a lot of failure. Like, you just have to be, all that's in your hands is to send that email, to send that application, to try, and the rest is not your job. It's not up to you to decide whether you're worth it, whether you're good enough, and it's not about you. There's like so many other people doing their own thing and trying their thing. And it's okay if it doesn't work out, right? There's just more experience to do the next thing better. So failure is a huge part of what I do, and I cannot get disheartened by it. I think all I can do is do my best, and that's what I'd encourage for you as well. So with that, I want to thank a bunch of people that have been completely essential to doing this work, especially all the field assistants and students that I've had. These are the latest. Padmanav just joined my lab as a JRF a couple of months ago. He's in Arunachal Pradesh doing some field work with sunbirds right now. And I've had a bunch of incredible collaborators and field sites that have supported this work. And I hopefully have time to take your questions. Thank you.
Thank you so much. That was a lovely talk. Am I audible?
Yeah, I can hear you.
Okay. We'll be, if that's okay with you, there are a lot of questions. I'm not sure if we'll be able to answer everything, but
should I
if
start
that's
with the
okay.
chat?
Yeah, sure.
Okay, so how does the physiology and energy expenditure mechanism of hummingbirds compare to bees, as they both feed on nectar and have a highly active lifestyle? That's a great question, and I don't know the answer offhand. I have actually been talking to someone in ISER Trivandrum who studies bees, Hema, and she is interested in things like this as well. So maybe we can collaborate and answer that a little bit better. But bees are ectotherms, and so they don't generate body heat on their own as much. They can do it with their muscles as a side effect when they're flying. But for the most part, they don't seem to, they seem to use external air and external sunlight and things like that to warm up. So I think their allometric scaling would be different than hummingbirds, but I don't know what it is offhand. That's a good question. I have a question that was sent to me directly. Would it be possible to have hummingbirds in India if we bring the eggs here? I wouldn't try to do that. There's been many situations of animal introductions to places where they were not native that have gone very badly. If you look in Australia, that's some examples. But here they'd also be competing with the sunbirds, and I think that's not a good thing to do if we can avoid it, to bring external non-native species to another place. Are the birds in that graph all the same species? So I think you're talking about the allometry graph. I'll just share it again. Sorry, one sec, let me go back. So I'm guessing that this is the one you were asking about. So in this graph, the individual points are individual birds, and the big colored points are species averages from that species. And so the colors are the species, and the bigger points are the species averages. So the next question is more CO2 emitted, the more energy spent? Exactly, yes. More CO2 means more energy spent. And you can estimate how much CO2 they would breathe out. So if you look at the ratio of carbon dioxide produced to oxygen consumed, that ratio is called a respiratory exchange ratio. And the closer it is to 1, the more likely they're burning carbohydrates. And the closer it is to 0.7, the more likely they are burning proteins and fats. So not only can you tell how much energy they're spending, you can also use that ratio to see what they're burning, what fuel they're burning to get that energy from. The next question is, what's the ratio of energy costs for hovering versus perching? Very good question. Hovering is 10 times as expensive as perching and sitting still. It's a very expensive activity, and hummingbird hovering is some of the most energetically expensive activities in the vertebrate world, in the animals with a backbone world. The name of the rat is the Nile grass rat. Thank you, Sonali answered that question already. The next question is, if a bird is able to survive by being an ectotherm, then why can't it continue being an ectotherm for a longer period and be an endotherm for less a period to save energy? Will it act as an ectotherm even during daytime or only at night? Very, very good question. First of all, most birds that we've studied don't seem to be heterotherms so far. There's only 3 groups that seem to be able to use torpor in birds by dropping more than 20 degrees Celsius. That's the nightjars, the hummingbirds, and the mousebirds. So there must be some cost, like you're suggesting, to being ectothermic at night. And I think there's also some hummingbird species where some individuals don't use torpor all the time, and they don't use torpor every night. So I think some of the costs are, one, that you suppress your immune system. And so if you're infected by something, you can't defend yourself against it. 2, you're completely useless in response to outside stimuli. You can't do anything if a predator comes for 20 or 30 minutes until you warm up, by which time you're probably dead. So if something finds you, which is more likely in the daytime, then you will die. And so I don't think hummingbirds would use it during the day if they ever had a choice, because they're sitting ducks. They can be found super easily by predators if they're just sitting there and can't respond. And then 3, there might be some, like, it seems to extend lifespans, but we don't know what other costs it has to the body. But I think the main ones are the immune function is suppressed and the not being able to respond. Those are pretty big. So I don't think many species have the ability to use torpor. Otherwise, we would see it more widespread as well. What's the relative size of these vital organs in the hummingbird, like comparing the size of the brain with the heart and the lungs? That's a good question. I think their hearts are big, and their pectoral muscle, which is the muscle that powers their flight, is huge. If you usually look in a bird, the pectoral muscle occupies like the top part of the chest. In a hummingbird, it's like the whole of the body cavity. If you dissect open a hummingbird, the pectoral muscle is just humongous. It takes the whole body cavity, and you have to take the pectoral muscle out to get to see any of the other organs. I don't know what the proportions are offhand in terms of numbers or percentages, but hummingbirds are pretty strange because they have this hovering flight. The next question, have similar studies been done on people or researchers staying in the Antarctic or Arctic regions for expeditions and how they fare to temperature changes day-wise or season-wise? Yes, there's a lot of people at the university I was at in the University of Alaska Fairbanks who actually study people in the military who go out in the Arctic and who are on expeditions which are like 12, 14 days. They eat huge numbers of calories. They're like consuming some 4,500 calories a day compared to 2,000, which we normal humans do. Because of the temperature, because of their activity levels. So there have been some studies, and it usually needs more energy if you're doing high-powered muscle activity in the Arctic, for example. Where I was staying, it was minus 20 degrees Celsius most of the winter, and sometimes it would go down to minus 40 degrees Celsius. There was like 1 or 2 days where it was even colder than that. So it's not a joke kind of temperatures. Is there a chance of something other, like another sense that these animals might have, like magnetoception that red foxes have? Do you mean hummingbirds specifically or heterotherms in general? Could you clarify that later on? I'll come back to it. Okay, why most of the birds lay 3 eggs in your experience when visiting a nest? How many eggs did you get most of the time? For hummingbirds, it's almost always 2. Sometimes it's 1, but usually it's about 2. And I think it's a trade-off between how much energy they can invest and how many offspring they can have. And there's a huge variation, like chickens can have many, many, many eggs, right? So it depends on the life history of the animal. And there's some animals that are called R selected and some animals that are called K selected. So R selected, I think, are the ones that have a lot of progeny or a lot of young over their lifetime, whereas K selected are very slow breeders. So like elephants and humans, we only have maybe 2 in our lifetimes, but we invest a lot of energy and a lot of time in taking care of them. Whereas the R selected species just like put out a bunch, like thousands, and hope that a few of them live. They don't do much paternal care or maternal care. So with the hummingbirds, they do invest time in taking care of them because they're incubating the eggs and the chicks for a few weeks after having them. So there is some investment, so they can't have that many if they're going to invest so much in each one. There's a lot of inspiring, wonderful, beautiful, informative. Thank you so much for saying all of those things. How can we connect with other bird enthusiasts? I'll let you answer that, Nafisa. I don't know what the best ways are.
Regarding that, with other bird enthusiasts, I think there are a lot of e-birding groups that most places have. So I think eBird's a good place where you can
Yeah,
interact
eBird is a great platform.
and also find other birders along there.
Yeah, so there's an organization in India called Bird Count India, which does a lot
Yes,
of this work. Some of the organizers of this course are involved with that, so Hale is. And there's a bunch of Facebook groups that you could probably find in your city for people who go birding every weekend, for instance.
There's one more question by Shivram. A hummingbird can flap their wings over 30 beats in a second. Their hearts were scaled to the size of a human's. Could we do the same?
I don't think so. I think it's there's some change when we get bigger. It's really, one is that we don't need as much energy per unit body mass usually. But also I think like, these broiler chickens, you've made them disproportionately have large hearts and disproportionately large, I don't know, their proportions are all weird and they don't do much. They just sit there. They don't fly anymore. They don't even walk much anymore. So I think scaling things disproportionate to the rest of the size of the organism usually doesn't work out very well. Another question is, why are they so brightly colored if they're so small and can be seen by predators and larger birds? It's probably a lot of it for mating. Sexual selection is a big criterion that people think is important for color. But also there's theories that it's just beauty that's being selected on. So if animals can see beauty, then that's what they're, I don't know. For the other senses, you meant specifically hummingbirds, but also curious about other ectotherms. So for hummingbirds, I think like many birds, they can see UV. I think that's important for them to be able to feed. They have a 4th color receptor. We humans only have 3 cones. They can taste very specifically. So like if you try to give them fake sugar, if you try to give them sucralose or aspartame, they know it's not sugar. They will not feed on it. So I think their taste sensors are acute and fine-tuned to nectar. What else? Their vision is very, very good. And they seem to respond very quickly. So their brains work very fast. I think they must think that we're like really slow, like giant animals. I think they see time faster than we do because they are preparing for their backstroke of their wing flap at the time that their front stroke is starting. People have looked at the neurons firing and they're able to like plan for the next move before the previous one has started. And so I think they see time differently, and that might be their superpower. Anushka is going to look for whether sunbirds are using torpor or not. Yeah, I'd love to know. So far in Africa, they found that they don't, but I really want to know in India if they do or not. Okay. Is that most of the questions? I don't know too much about sparrows. I know that they're decreasing in India and it's not great, but they're also not native here. So yeah, there are no hummingbirds in India. Like I said, they're only in North and South America. So there are sunbirds here, which are evolutionarily distant and ecologically convergent, but we don't have hummingbirds. Sorry, I forgot to not stop sharing my screen. Yeah.
No worries. Thank you so much for this talk. This was lovely, and this was a very interesting session. Every bit of it was amazing. Thank you so much. And thank you
Thank you.
to everyone who's attended. Thank you so much, and thank you to everyone who's attended this live session as well. I hope you all had a nice session. Thank you so much.
Thanks. Have a good day and evening and night.
You can stop. We can. Yeah. We can. Oh, the recording, is it stopped? Okay. I think the recording has stopped now.
Yeah, it's hard to tell.
I'm not sure if this. Oh, this was a lovely session, by the way. Thank you once again.
Yeah, I'm glad you invited me again.
I
Should I leave the session now?
yeah, most welcome.
Okay,
Thank you so
thank
much.
you all. Thanks, Nafisa. Bye.
Bye bye.